So first I am going to do my starch agar plate. I already kind of drew on it, but before I videotaped it. So I'm gonna sterilize my loop in the micro incinerator, and then I'm gonna grab this, make sure it's mixed up so I can grab all the bacteria. And then next I'm gonna open it with the one-handed aseptic technique, and then I'm gonna sterilize it, and then I'm gonna grab my loop. It's already been cooled. Grab the bacteria out, sterilize the tube, the source culture broth, and I'm going to close it, put it back, and then I'm going to grab my loop, and I'm going to streak the plate like this, and then I'm going to close it, incubate it upside down, and then after it's been incubated for about 24 to 48 hours, the next lab, I'm going to grab my iodine solution. I'm going to put a couple drops in there, and then it will indicate my results. The next plate is the case and agar plate. I'm gonna sterilize my micro incinerator, let it cool, grab my source, mix it up, sterilize the tip of it, grab my cooled down loop, grab some culture like this, grabbing the culture, taking it out, sterilizing it, making sure I'm doing all aseptic techniques, putting it back, grabbing my loop, and what I'm going to do here is I'm going to make a single streak like that. Next, I'm going to do the tryptophan broth, which is the indole test. I'm going to sterilize my loop, grab my culture, make sure it's mixed up, sterilize the tip of the culture, grab my cooled loop, put it back, put it in the culture just like this, take it out, sterilize it again, and then I'm going to take my loop, and then I'm going to open up the tryptophan broth aseptic technique, obviously. Make sure this is sterilized by that before I inoculate, and once I inoculate it, I go like this, take it back out, sterilize it again, close it back up, and then sterilize my loop, and then you're done. Incubate it, close it up, incubate it in one of these holders. Next, we've got our urea slants right here, and I already drew on them, but I will go over that again. We're going to sterilize our needle. We're going to grab our culture. We're going to take it off aseptically. We're going to sterilize it, and then we're going to grab our cooled needle, take some bacteria off just like this, not too little, not too much, sterilize it, put it back on. We're going to take our urea slants. We got a side and a face view. So first we're going to take it off aseptically, and then we're going to sterilize the tip, and then we're going to take our cooled needle with the bacteria on it, and then we're gonna go like this. You're gonna dig into that urea, and then once we're going back up on the slant, you're gonna come back up about here, and then you're gonna streak on that slant. For the next test is the catalase test, and for this one we're not actually going to be using a needle or a loop. We're going to be using a wooden stick. So let's say this is our wooden stick, but first we're going to have a clean slide that's been cleaned, sterilized, has no bacteria on it, no cross-contamination, nothing, and we always hold it by the sides. So with your marker or with your pen, whatever, you're going to make a loop, or you're going to make a circle indicating where you're going to be smearing that bacteria. And then next, what you're going to do is you're going to grab hydrogen peroxide from your cabinet, and you're going to put a drop of that in the center of the circle, and then you're going to obtain the wooden stick, and you're going to grab your culture, you're going to open it aseptically, you're going to sterilize it, and you're going to grab your wooden stick that's been already it's cleaned, it's not been used anywhere. You're going to grab the stick, and then you're going to grab a little bit of culture, and you're going to sterilize that, and then take it and close it back up, and then leave that to the side. And then what you're going to do is you're going to apply that culture onto the hydrogen peroxide, and then that will indicate whether it's positive or negative, and you will observe for the formation of bubbles. For this next video, we are going to be doing the cotton swab oxidase test, and we're not going to be using a needle or a loop for this one. So what we're going to do is we're going to grab our source culture, and we're going to have our cotton swab ready already out of the package. We're going to take our source culture, take it off aseptically, sterilize it, let it cool for just about seconds or however long it takes. Take your cotton swab, grab just a teeny bit of culture, and then sterilize it again, and then put it back where it belongs. And then in your reagent cabinet, you're going to take a pipette. You're going to grab about 1 to 2 drops, and then you're going to place it on the cotton swab, and that will indicate whether you've got a positive or a negative test. So next is the Durham sugar, and this one's pretty simple inoculation wise. So you're going to take your loop, you're going to sterilize it, let it cool, grab your source culture. You're going to obviously take it off aseptically, sterilize that, the tip of that, let it cool, grab your loop, grab just a teeny bit of bacteria on that loop, not too much, not too little, and then you're going to sterilize that again. And then you're going to take your Durham tube, open it up aseptically, sterilize it, let it cool for a bit, and then you're going to take your bacteria, looped bacteria, and then you are going to inoculate it. And then what you're going to do is you're going to mix that up just a bit in there, and you're going to sterilize it again, put it in, put the cap back on, and then incubate that just like that. So for this next test, we got the MR and the VP test, and they're pretty much, they go together, I guess you could say, if you read the procedure. So what first we're going to do is we're going to take our loop, and then we are going to sterilize it, let it cool, grab our source culture, take it off aseptically, sterilize the loop, the tip of that, and then we're going to grab our sterilized and cooled off loop. We're going to grab just a bit of culture, sterilize that culture tip again, and then we're going to set it off to the side. Let's do the MR test first. So what we're going to do is then we're going to take our loop, open this up aseptically, sterilize it, grab our culture bacteria. We're going to inoculate it just like that, mix it up, and then we're going to sterilize it again, put the cap back on, and then let it sit and incubate that just like how it is in the picture. And for the next test is the VP test. What we're going to do for that one is we're going to sterilize our loop, and then we are going to sterilize our source culture while that loop is cooling, and then we're going to take just a bit of bacteria, and then we're going to sterilize it again, and then let that sit on the side, and then we're going to grab our VP test, sterilize the tip of that aseptically with the one-handed pinky thing that we learned in the first couple weeks of class, and then we're going to sterilize the tip of that, and then we're going to grab our loop with the bacteria on it. We're going to inoculate that just like that, let that mix around, and then we're going to sterilize it again, and then we're going to close that up and then incubate it just how it is like that. After this methyl red has been incubated for about 48 to 72 hours, we're going to take our methyl red solution and then add about 20 drops into there. And then making sure you're not spilling any of that, all that good stuff. And then after you add those 20 drops, you make sure you swirl the tube frequently so it can mix in real well. And then after the VP test has been incubated for about 48 to 72 hours, we are going to first grab our 10 drops of alpha napithol, I believe it's called, and then we're going to add 10 drops into that. And then you're going to make sure that you're stirring it very well. And then after that, we are going to add the 10 drops of KOH. And then you got to mix it well without stirring it. And then it is very important to incorporate the mixture. So you got to make sure to be stirring it real well. And then you place, after you put the drops of KOH, you place the VP test into the incubator for about 30 minutes, returning every 5 or so minutes to mix that up. And that's very important to get to get accurate results. So make sure to do that. This next one is the nitrate broth, and I'd probably say this one's my favorite just because the results are so confusing. Anyway, so we've got the nitrate broth. You're going to sterilize your loop, let that cool, grab your source culture, take it off with your pinky, sterilize the tip of that, and then grab some of your grab your loop, grab some of that culture, just a bit, not too much, not too little, as we learned in the previous labs. And then you're going to sterilize that again, put your lid back on, and let that rest. Now we're going to take your nitrate broth, and you're going to take it off again, aseptically, sterilize that tip, and then you're going to take your loop, and then you're going to mix your bacteria into the nitrate broth, just like that, and then you're going to sterilize that tip again, and then put it back on, and then you're going to incubate it. After your nitrate broth has been incubated, you are going to grab your sulfanic acid, and then that is considered nitrate 8. You're going to add 20 drops of that into there, and then you're going to wait a bit, and then you're going to add another 20 drops of the alpha naphthylamine, which is the nitrate B, which is this one right here, and then you're going to add that to there. And if the broth turns red, then that's a positive reductase result. And one thing to note is it's suggested that you allow the broth to stand for a few minutes, mixing it frequently, so that reagents can penetrate adequately. And then the only then is when you should make the decision on the color change. And then that is when, and then if the broth does not change, so if there's no color, you're going to add a few particles of the zinc. Sorry, my printer kind of messed this up, but you're going to add zinc dust with a little toothpick, and then you're going to add that into there. And then if the broth turns red, then that is a negative reductase result, and if it does not turn red, that is a positive. And that does get a bit confusing, but that's why you got to make sure that you study these ones. For this next one, you've got the citrate slant, and this one's pretty cool too, just because of the colors. And then what you're going to do is you're going to be using a needle. You're going to you're going to sterilize your needle, take it out, sterilize the tip of this, obviously with the aseptic technique. Sterilize it, grab your needle, grab just a smudge of bacteria, and then sterilize it again, and then put that back on, let that sit to the side. You're going to grab your citrate slant, and then you're going to sterilize that tip, and then you're going to grab your needle. You're going to stab it, and then when you're going back up, you're going to streak the slant of that, and then once you're done with that, you're going to sterilize it, close it back up, incubate it. So this next one is the motility deep needle, and this one is very important. You got to be very careful with this one. But you grab your needle, you're going to sterilize it, grab your source culture, you're going to sterilize the tip of that, obviously using aseptic technique. When you remove the cap, and then you're going to grab your cool needle and then grab a smudge of bacteria, and then once you do that, you are going to sterilize it again, the tip of that, close it up, put it to the side. Next, you grab your motility. Now this is very jelly, so you gotta be careful with that. You grab this, sterilize the tip of it, obviously removing aseptically, and then you're going to grab your needle. But what you do for this one, you gotta make sure that you inoculate your motility tube by stabbing it directly, and then you do not touch the bottom of the deep with the needle, and then you withdraw your needle along the exact same line, so like this. You gotta be very careful. So once you stab it, you go back up just exactly how you stabbed it. Try not to move your needle. And then after that, you are going to sterilize your needle, sterilize the tip of that, and then you're going to close it and then let it incubate, and then you're done with that one. For this next one, we got the TSI slant. And the TSI slant, you take your needle, it's also a needle, you're going to sterilize it, let it cool, grab your source culture, and then you're going to take that, and then you're going to sterilize the tip of that, grab just a bit of culture, sterilize the tip, close it back up, obviously doing this all aseptically, let that sit. Take your TSI slant, sterilize the tip, grab your bacteria, and what you're going to do is you're going to stab that, and then once you're going back up, which right here, you're going to streak along the slant, just like that. And then once you're done, you're going to sterilize the tip of that, let that go, incubate it, sterilize your needle, obviously, and then you're done. You're going to incubate it and then wait for results the next lab.